Hello, good morning. Scanning electron microscopy, SEM. It is a type of microscope which uses the electrons to illuminate a specimen and create an enlarged image principle. The signals used by electron beam scanning electron microscopes to produce an image result from interaction of the electron beam with the sample. Various types of signals are produced. When high-energy electron beam strikes the sample, some electrons are scattered. See first this diagram. When electron strike on the sample specimen, some electrons are scattered, and X-rays and secondary electrons. These three types of electrons. So some electrons are scattered due to elastic scattering, the backscattered electron. Some of the electrons are knocked off from the surface, secondary electron. Some of the electrons penetrates deep into the inner cells of the sample atoms to knock off the inner cells electrons, due to which characteristic X-ray are produced. See, these are scattered electrons, and these are scattered electrons, backscattered electrons. Some electrons are knocked off from the surface, and these electrons are these secondary electrons. And third one, electrons which penetrates into the inner cells of the sample and at a sample's atom to knock off the inner cells electron, due to which the characteristics penetrate the inner cells of sample atom to knock off the inner cells of the electron. The X-rays, three types of electrons in the same instrumentation: scanning electron microscope, electron gun, anode, condenser, scan coils, objective lens sample, and this is the secondary electron detector. Electron gun contains tungsten filament. 1 to 30 kilovolt accelerating voltage is applied to produce the electron beam. Anode, it is positively charged and attracts the negatively charged electrons, and ultimately this anode will accelerate the velocity of the electrons. Electron beam is controlled by anode, condenser lens, and objective lens and lens. These are electromagnetic lens. Condenser lens, this lens will focus the electron beam towards the objective lens. Objective lens, the lens which again flows the electron beam toward the sample specimen. Sample specimen. Sample preparation. The sample preparation are occurred by flowing ways: cleaning, drying, dehydration, fixation, sample coating, acetone alcohol. This microscope cannot be used to image living cells. Detection in SEM. These three types of detection is possible. Secondary electrons. Secondary electrons are most abundant in the case of SEM and detected by the suitable detector. Eberhard only ET detector is most commonly used electrons in SEM. ET detector contains base grid framework network which is positively charged to attract secondary electrons. Secondary electrons detected by ET detector, Eberhard only. ET detector contains, okay, I have already attract the secondary electrons. The secondary electrons detected by ET detector will produce the basic image of same. Backscattered electrons. Energy of backscattered electrons are higher than secondary electrons, so ET detector of varying voltage of difference between them. X-ray detector. Inside the sample holder, X-ray detector is placed and it will detect the X-rays emitted by sample. The detection process is also known as EDX or Energy Dispersive Analysis process. Application of SEM for investigation of virus structure, 3D tissue imaging of insect, spore, other microorganism or cellular component visualized. Geologists often use SEM to learn more about crystalline structure. Industries including microelectronics, medical device, food processing, all use SEM as a way to examine the surface composition of component and product. Okay, it's all about scanning electron microscope. And this is the application, detection, sample preparation, and instrumentation of SEM. This is the principle of the SEM. Okay, thanks for watching. And request you all like, comment, share, and subscribe. Thank you for your patience. That is now. Bye bye. See you again.